Welcome to part 9 of this series of slideshows that describe and discuss the mammalian stress mechanism that was predicted by Hans Selye. Recent advances in the understanding of turbulence provide revolutionary insights to the nature of mammalian physiology with implications that exceed the bounds of medicine. This presentation will discuss the nature of pipe flow turbulence and its relationship to vertebrate physiology. Turbulence was the greatest mystery of physics and engineering until it was recently discovered to have underlying order that can be described by mathematical equations. This led to the founding of a new branch of science known as chaos theory. Turbulent equations are called Mandelbrot equations, or fractals. The pictures above are examples of computer-generated fractals. There is similar underlying order in cloud formations, tree limbs, broccoli stalks, ferns, irregular coastlines, weather patterns, craggy mountain ranges, lung structure, heart rhythms, and pipe flow. Turbulent equations also explain how DNA efficiently stores anatomical shapes and forms that characterize complex animal and plant structures, as illustrated by the Barnsley's fern fractal equation on the right. In 2004, Belgian mechanical engineers used laser photography of water pipe flow to confirm mathematical predictions of turbulent pipe flow patterns. Turbulence appears in the form of bright red jet streams that move forward along the inner walls of the pipe. These force slower moving elements of the water toward the center of the pipe, where they move backward. As acceleration increases, the turbulent patterns temporarily persist and then suddenly transform to alternate turbulent patterns. This explains why turbulence induces pipe flow resistance and how it can dislodge rust from the inner surface of old pipes. At peak turbulent intensity, turbulence rings the inner surface of the vessel. Pulsatile blood flow operates at the threshold of peak turbulent intensity because this mobilizes particulate deposits from the inner surface of arteries and prevents atherosclerosis. The turbulence generates lateral forces that press on the inner wall of the vessel. These forces explain the palpable pulse and blood pressure measurement. Turbulent relationships are exponential, unstable, and powerful. Small changes can drastically alter turbulent patterns. This is called the butterfly effect, as when a butterfly flapping its wings in China causes a tornado in North America. Gun design illustrates the unpredictable interaction of turbulent variables. Gun design is a process of trial and error that achieves practical combinations of gunpowder chemistry and quantity, ambient temperature and humidity, cartridge size and shape, bullet size, shape and mass, barrel diameter and length, metal characteristics, and so forth. Altering any of these variables, and especially altering two or more variables simultaneously, may produce a gun that is unreliable, ineffective, or even dangerous to use. Pipe flow turbulence is affected by pipe length, diameter, curvature, bifurcation, imperfections, temperature, particulates, and the rate of acceleration. Turbulent lateral forces generated by pipe flow turbulence might be compared to the Bernoulli principle, where turbulent flow over the curved surface of an airplane wing generates powerful lifting forces that enable airplanes to fly. Such forces can explode or collapse pipes. Plumbers call this water hammer. Most plumbing systems in ordinary homes include a trapped column of air that cushions water hammer forces. Turbulence is the sole cause of pipe flow resistance, which is called viscosity. In medical literature, blood viscosity is customarily called systemic vascular resistance. Most common liquids, fluids, and gases such as air, carbon dioxide, water, milk, Kool-Aid, gasoline, oil, and so forth are called Newtonian fluids because they exhibit exponential increases in viscosity with increasing acceleration. Blood and cornstarch solutions are non-Newtonian fluids because they exhibit exponential decreases in viscosity with increasing acceleration. Non-Newtonian fluids are characterized by the presence of particulates that inhibit pipe flow turbulence. As blood accelerates, the unique shape and size of mammalian red blood cells causes them to spontaneously organize themselves into stacks that are called aggregate patterns in research literature. These red cell formations abolish turbulent flow patterns and eliminate flow resistance. Because of this, mammalian blood exhibits laminar flow during systole. Laminar flow enables extreme cardiac efficiency comparable to electrical superconductivity. Cardiac contractility is converted into pure kinetic energy with nearly 100% efficiency because turbulent flow resistance is absent. This explains how the mammalian heart can effortlessly increase its output by a factor of 10 during intense exercise, why athletic conditioning has minimal effect on heart size and weight, and why the exercise capacity of mammals is superior to that of reptiles. Hemoglobin encapsulation distinguishes vertebrates from invertebrates, and each class of vertebrates appears to be characterized by its own distinctive red cell morphology. This remains totally unexplained. Conventional theory assumes that the primary purpose of red cells is to deliver oxygen to peripheral tissues, but encapsulation does not improve the oxygen carrying capacity of hemoglobin or tissue delivery of oxygen, and red cell mass is far greater than needed for effective oxygen transport. Red cell morphology alters vertebrate blood turbulence to facilitate environmental adaptation. Mammals evolved directly from reptiles, so they are closely related, but their red cells are dramatically different. Reptilian red cells are large, nucleated, and elliptical. Reptiles allow their body temperature to fluctuate with environmental temperatures, and their red cells exaggerate systolic blood turbulence to counteract atherosclerosis acceleration due to lipoprotein solidification at cool temperatures. This explains why reptiles have limited exercise tolerance and inferior cold tolerance. However, they require far less food than mammals. Mammals maintain their body temperature at the threshold of lipoprotein liquefaction so that lipoproteins blend with liquid blood elements and do not affect blood viscosity. This eliminates the problem of accelerated atherosclerosis caused by lipoprotein solidification at cool temperatures and enables them to employ small biconcave red cells that inhibit systolic blood turbulence and optimize cardiac efficiency and exercise tolerance. This enables mammals to thrive and function in cool temperatures, but it exaggerates food requirements and heat intolerance compared to reptiles. In mammals, anemia exaggerates blood turbulence, which impairs cardiac efficiency, undermines exercise tolerance, and exaggerates atherosclerosis resistance. Severe anemia, which most often occurs due to chronic renal failure, can paralyze hemostasis and mimic disseminated intravascular coagulation, but the cause is different. Hemostasis is restored with erythropoietin or transfusion with packed red blood cells to restore normal red cell mass. Excessive red cell mass, or polycythemia, inhibits blood turbulence, exaggerates blood coagulability, and accelerates atherosclerosis. Within limits, it enhances cardiac efficiency and exercise tolerance, which explains why athletes treat themselves with erythropoietin to enhance athletic performance, which is called blood doping. Polycythemia can also be caused by alcoholism, overenthusiastic erythropoietin treatment for anemia, and excessive transfusion with packed red blood cells. It increases the risk of DIC, and in severe cases can trigger DIC with devastating consequences. It's time for another break. The next slideshow will discuss the medical implications of mammalian blood turbulence. Thanks for listening.